Cellular Respiration by Matt Danowski
and Matt Duke.
and maybe Robbie.
Cellular respiration is the process where glucose and oxygen are used to make energy for a cell.
Glycolysis takes glucose molecule and breaks it down into two pyruvate molecules in the cytoplasm of the cell. It also creates a net worth of 2 ATP.
During the first step of glycolysis, an ATP molecule is hydrolyzed to attach a phosphate group to the end of the glucose molecule. This becomes glucose 6-phosphate. The molecule is then rearranged to become fructose 6-phosphate. The same process then occurs for the other side of the glucose molecule. The molecule is now called fructose 1,6-bisphosphate.
The backbone of the fructose 1,6-bisphosphate is split in two, creating a glyceraldehyde 3-phosphate and a dihydroxyl acetone phosphate. The dihydroxyl acetone phosphate is then converted to another glyceraldehyde 3-phosphate.
NAD plus is reduced to form NADH from G3P by giving up one hydrogen and 2 electrons. Alright, so the next step, G3P combines with an inorganic phosphate, and this is an unstable molecule that is called 1,3-bisphosphoglycerate. The 1,3-bisphosphoglycerate gives up its phosphate group to ADP, forming ATP.
This is called substrate level phosphorylation. The molecule of 3-phosphoglycerate rearranges to a structurally different isomer 2-phosphoglycerate. 2-phosphoglycerate gives up one proton and one hydroxide ion, which combine to form water. The resulting molecule is phosphophenols, or PEP. PEP is very unstable and breaks down. The energy released is enough to transfer one phosphate group to ADP, forming another molecule of ATP.
So, the original glucose molecule has been broken down to 2 pyruvate molecules, each with a 3-carbon backbone. In summary, glycolysis produces 2 NADH molecules, 2 ATP in net, and 2 pyruvate molecules for each molecule of glucose entering the reactions. Next step, pyruvate oxidation.
Yeah, bud. At this point, the pyruvate molecules produced by glycolysis enter the matrix, the mitochondrial matrix.
Coenzyme A attaches to the acetyl group of the pyruvate molecule, which enables it to be oxidized. At the same time, NAD plus is reduced to form NADH. The remaining carboxyl group is removed as waste in the form of CO2. The resulting product is now acetyl-CoA.
When energy is needed by the cell, the acetyl-CoA enters the Krebs cycle to continue aerobic respiration.
Next step, Krebs cycle! In the first step of the Krebs cycle, acetyl-CoA reacts with oxaloacetate to make citrate. Citrate changes its shape to make isocitrate. Through a redox reaction, isocitrate oxidizes, which means it loses an electron, to become alpha ketoglutarate, which can be found here in the Krebs cycle. Alright, in the next part, NAD plus gains an electron, and at this point it is converted to NADH. which can be found here. Also in result, there is waste of CO2, and then we're on to the next step. So alpha ketoglutarate binds with coenzyme A and is oxidized by NAD+ to form succinyl-CoA. So to illustrate this, we'll do a picture over here. So we're going to draw a little man. Look at that. It's pretty strong. Alright, he's got some weird arms, but that's okay. So, binding, so this man is going to represent alpha ketoglutarate. So we have NAD+ in this guy's arm. It's binding to him, and he also has coenzyme A. So now, instead of his name being alpha ketoglutarate, we can now call him succinyl-CoA. Now, at this point, CO2 is released as well. There you go. So you remember our friend succinyl-CoA. He's a pretty important guy. He helps us form succinate, which is also the step in which we get our first ATP. So that is pretty important. To do so, succinyl-CoA has to lose its CoA, and through substrate phosphorylation, we have GDP transferred to make GTP. Now the relevance of this really is that this process just gives succinyl-CoA the energy it needs to take ADP and add inorganic phosphate to make the ultimate goal of all cell respiration is ATP. So now that we have our succinate that has officially given us our first ATP, we can now turn succinate into fumarate. So fumarate is made by FAD, important molecule here, and it is oxidized to form FADH2. Now, in this process, we get fumarate. So now we have some nice water. Right? We got some sharks in it. No, just kidding. There's no sharks in it. Anyway, so this water is added to our fumarate, and now this fumarate is transformed into what many call malate. And at this point, we've almost come full circle, so just bear with me. We've got one more step. So, our malate from the last step undergoes a redox reaction with NAD+ to produce NADH. And we also get the beginning cycle, the beginning molecule of the cycle, which is oxaloacetate. Now that we have this, we can start the Krebs cycle again, and the Krebs cycle does happen 2 times. So after this, the Krebs cycle happens again. So in total, the Krebs happens 2 times. Alright? So in the end, we end up with 2 ATP, 6 NADH, and last but not least, we have 2 FADH2. The next step is ETC and it is found in the inner mitochondrial membrane.
Yay!
That's how you spell membrane. The first 5 components of ETC are NADH, dehydrogenase, as well as succinate dehydrogenase, and cytochrome B C1 complex, cytochrome oxidase, and finally we have oxygen. And all these components increase in electronegativity. The other 2 components are 2 mobile electron carriers. One of them is a funny word, but ubiquinone, or UQ. And we also have cytochrome C, which can also be known as Site C. ETC aims to make ATP during cellular respiration, and this step out of glycolysis, pyruvate oxidation, the Krebs cycle makes the most ATP. As an overview, we're going to draw the inner mitochondrial membrane and just draw in some complexes that will represent the major components. So we have complex 1, we have complex 2, 3, 4. Let me just put some of this in. And the 5th one, to extend this, is ATP synthase. This one's pretty important because this is where we get a product of ATP in the end. So in order of electronegativity, we have complex 1, which is NADH dehydrogenase, complex 2, which is succinate dehydrogenase, complex 3, which is cytochrome BC1 complex, complex 4 is the cytochrome oxidase, and oxygen finds its way in the system as a terminal electron acceptor.
The proteins in the ECC are set up in order of increasing electronegativity. The first component is a weak electron acceptor, and the last component is a strong electron acceptor. Electrons are passed from one component to the next along the membrane. Initially, NADH binds to NADH dehydrogenase, where it is split up into NAD+. A hydrogen ion is pushed into the intermembrane space, and an electron is passed along the chain. FADH2 binds to succinate dehydrogenase and follows a similar process. UQ, mobile electron carrier, carries electrons from NADH dehydrogenase and succinate dehydrogenase to cytochrome BC1. Then Site C carries the electrons from cytochrome BC1 to cytochrome oxidase. Because FADH2 passes through one less electron carrier in the chain, it creates one less hydrogen ion in the inner membrane space, and therefore creates one less ATP. So NADH produces 3 ATP.
So 3 ATP, and FADH2 creates 2 ATP. The next step is chemiosmosis. And this can only be achieved after the electrochemical gradient has been created by ETC. So now we're going to redraw our picture from the last slide. So we have our inner mitochondrial membrane, and we have our ATP synthase. There it is. Make that a little smaller. So previously we said that the hydrogen ions came through this pump and mingled around and were combined with the oxygen that had received 2 electrons to make our water. Okay, H2O. So these flow of hydrogen ions provide energy to make a bond between ADP and pi. A resulting product is ATP. And this specific process is called oxidative phosphorylation. So here's the summary. So the 6 NADH molecules and 2 FADH2 molecules are placed within ETC, previously explained, and through that we yield 22 ATP. Combined with glycolysis and pyruvate, oxidation, and the Krebs cycle, we get a total of 36 to 38 ATP. So that's all, folks. We've successfully explained cellular respiration. And get well soon, Robbie and Matt. You will be missed in biology.